Today we'll be talking about the Zaire Ebola virus. Here is a picture of the Ebola virus strand. So what is the Zaire Ebola virus? It is a virus that was discovered in 1976 and experts believed that it is originated from fruit bats. It is one of many hemorrhagic fever, which is an illness that is caused by several families of viruses. It is contracted through physical contact of the fluid of those who are infected. Those that are infected with the disease experience fever, vomiting, muscle pain, fatigue, weakness, and much more. So if any of you guys are experiencing these symptoms, you might want to see a doctor about it. Some proteins that are affected are nuclear protein, viral proteins, and glycoproteins. But the one we are focusing on today is the membrane-associated protein GP1. This is the molecular structure of the glycoprotein that is infected with the virus. The region in red is the affected GP1, and the region in green is the affected GP2. So what exactly is GP1? GP1 is a viral glycoprotein that has 676 amino acids and 2,029 nucleotides. It is essential for infectivity since it's the only viral protein expressed at the surface. First enters the cell by tricking the cell membrane into swallowing the virus. The process is called macropinocytosis. This is triggered by glycoprotein. To visualize the cell swallowing the virus, just picture someone eating a sandwich. When the virus enters the cell, it will be in a package called the endosome. In order for the virus to take over the cell, the virus must somehow escape the endosome and into the cytoplasm of the cell. This is when MPC1 comes in. MPC1 is attached to the glycoprotein. It binds to the receptors of the endosome and tricks the endosome into opening up its membrane. Once the endosome opens its membrane, the virus will releases its genetic material into the cell's cytoplasm and uses the cell's replication machinery to replicate itself. Essentially, the virus takes over the cell and turns it into a factory to make more copies of itself. Eventually, the infected cell will release copies of the virus and they will start targeting other immune system cells and other healthy cells like liver cells.
GP1 undergoes alternative splicing by NPC1. Niemann-Pick disease, type C1, is a protein-coding gene involved in cholesterol transport and storage. It is important for viral entry for GP1. Studies have shown that by preventing the binding of NPC1 to GP1 allows it to inhibit the viral infection, therefore suggesting that NPC1 can be a major contributor to an Ebola antivirus viral drug. This is an image of the GP binding to NPC1. As you can see, the Ebola virus is attached to the GP protein, which is attached to NPC1, which is then attached to the cytoplasm. You can see how the virus is then let into the cell through NPC1. NPC1 is essential for the entry of the Ebola virus. So what exactly is connecting GP and NPC1 together? It is the protruding loops between the two proteins. However, Loop 2 plays more of an important role when it comes to the binding of both proteins. How is GP associated with Ebola? The expression of GP1 can reduce the levels of the host cell's surface protein, which means that it plays an important role in viral entry. The study of the structure and function of GP can be used to design the inhibitors that can prevent cells from being infected by the disease.
Cloning GP1 can reveal the limit of antibodies in humans, and it can also help expand our knowledge on viral entry prevention methods. To express our GP1 protein, we use the polymerase chain reaction to clone our gene. We use primer 3+ to help us pick suitable primers for the PCR of GP1, and then we use primer blast to confirm the functionality of our primers. We expressed our gene in E. coli and transformed the bacteria. Once the E. coli is transformed, we can use it to induce the expression of our gene. We were able to harvest, lyse, and purify the protein. This proves that our gene can be expressed. The purification process of GP1 involves the usage of the PMAL-C5X plasmid to label our GP with a maltose binding protein. Affinity chromatography is used to purify our gene from the lysed E. coli cells. We then visualized our gene on a SDS page gel.
created using Powtoon.